Good afternoon, AP biologists. So good to join me today on this journey through meiosis. For your do now, hopefully you were able to get through the gizmo and practice some very important causes of genetic variation and see the result on the phenotype, which is physical characteristics, from the genotype, which is the genes on the allele. Continuing on that same thread, we're going to go through two FRQ questions today. And the first one, we'll just dive right in. A research team has genetically engineered a strain of fruit flies, just like what we saw in our do now, to eliminate errors during DNA replication. The team claims that this will eliminate genetic variation in engineered flies. A second research team claims that eliminating errors during DNA replication will not entirely eliminate genetic variation in the engineered flies. Describe one mechanism that could lead to genetic variation in the engineered strain of flies. So, thinking about what the prompt is saying, it's talking about genetic variation, but it is saying that if we're not talking about genetic variation during DNA replication. So what genetic variation will we be talking about during DNA replication? Well, if you're thinking about DNA replication like we learned in Module 10, you're immediately thinking about what class? Yes, you all yelled out mutations. You're like, oh yes, mutations cause variation. And they do, because they give us those new sequences. Wow, look at me, right? Those new sequences, but we can't say mutations because it says we've eliminated errors in DNA. So then what is another mechanism? Well, you should learn what you did in your do now was what? What did you do in your do now? Yell it out. Yes, you said crossing over, or synapsis. So crossing over is definitely one of the ways that you get new alleles on new chromosomes. So that's when you swap alleles between homologous chromosomes to get new combinations of traits. So just like in our do now, right, we had crossing over led to different combinations, either body color or wing size. So it's it used to be that if you had vestigial wings, you had a black body, but now you can have a brown body and vestigial wings. So that's all from crossing over when those genes are on the same chromosome or linked. Great, so one source of genetic variation, boom, I've nailed it. There are so many other ones and you probably can yell them out right now. Yes, you did yell out random fertilization, independent assortment. What else could you have said? Okay, those would probably be the ones that you would have said. Meiosis, which is producing new combinations of alleles, which is independent assortment. Or the random combinations of alleles present in haploid gamete cells. Or you could have said random fertilization, right? Which is just exactly what it sounds. When you have a random sperm and a random egg see each other across the fallopian tube and make a baby. Those are the ones you should have said. These are some other ones in your key that you could look for. A lot of them we haven't talked about, like viral infection inserting DNA, but we talked about in our last lesson. All non-disjunction, so when the cell doesn't split properly. All of these things are possible answers to this question. So lots of options to get 1 point. Please reach out if you have any questions at this time. The other part of this lesson, other than genetic variation, is comparing meiosis and mitosis. So in many organisms, it's a part of sexual reproduction, meiosis. So that's how you make gamete cells or haploid cells. So what are 3 differences between meiosis and mitosis? So in this kind of case, you might just want to think about mitosis. What do we have at the end of mitosis? Ooh, I thought that was going to write. Mitosis. So what are you going to yell out? At mitosis, we end up with, yes, I hear you, 2 identical daughter cells. Ooh, and are they diploid or haploid class? Yes, they are diploid. Good job. That means you have 46 chromosomes, or 2n of number of chromosomes. And that would be what we end up with in mitosis. How about in meiosis? Just yell it out. Yes, it is 4 different, right? Because they have genetic variation, which we just talked about above, right? Meiosis crossing over an independent assortment give us that genetic variation, so that we can have 4 different cells. And those cells are what? Yes, I heard you, haploid. Nailed it. And that means N, or half the number of cells. So when we are done with fertilization, when an egg meets a sperm, we have back to diploid numbers. So each egg and sperm have half the amount of chromosomes. So when they come together to make a baby, the baby has a full number. What are some other differences between meiosis and mitosis? If we're thinking about the number of divisions, yell it out. What is the difference? Yes, mitosis has 1 division. Meiosis has 2 divisions. Nailed it. That's because it has 2 divisions. That's because we need to divide not only the homologous chromosomes, but then also the sister chromatids. So 2 divisions, but remember, there's no DNA replication between division. So DNA replication still happens in S phase, which is before meiosis, during interphase. So between meiosis 1 and meiosis 2, there is no DNA replication. What other things can you talk about? Well, in meiosis, above, we talked about crossing over. We talked about independent assortment, because those both happen in meiosis, but do not happen in mitosis. There's none of that, because they do not have the swap of crossing over of homologous chromosomes. And then finally, what is the purpose of each? Yell it out. What's the purpose of mitosis? Yes, you said growth, you said repair, and then you said asexual reproduction, just like your aloe plant, which is doing that right now. But what about meiosis? What's the purpose? Yes, to form gametes, so eggs and sperms for sexual reproduction. Great. So that way you can get those genetic variation. Well, that's all I have for you here. Below, make sure that you finish the independent practice questions, which kind of look similar to what we just talked about. And then make sure you grade yourself like you did in Module 9. So just like look at the answer key on the PowerPoint and then give yourself a grade however long it took you to get done in 15 minutes. So set a timer, and at the end of 15 minutes, let's say you don't get to the last one, well, that's a 0 out of 2, just like it would be on the test. So I hope that you all have a great day, and I look forward to seeing you next time.